Nucleic acids are one of the naturally occurring chemical compounds or large biological molecules and are therefore polymeric macromolecules, which is imperative for all forms of living organisms. They are capable of being disintegrated to yield phosphoric acid, sugars, and a mixture of organic bases like the purines and pyrimidines. Nucleic acids are the communication channels and the main information carrying molecules of the cell, and by overseeing and directing the process of protein synthesis, they determine the hereditary characteristics of every living organism. The two main classes of nucleic acids are deoxyribonucleic acid, that is DNA, and ribonucleic acid, that is RNA. Both the classes of nucleic acid are of utmost importance to derive the innate characteristics of life forms. DNA is the master blueprint for life and comprises genetic material in all free living organisms and most viruses. RNA is the genetic material of certain viruses, but it is also found in all living cells, where it plays an important role in selective processes such as the formation of proteins. This chapter will deal at length with the chemistry of nucleic acids and will describe the structures and properties that allow them to play the role of transmitters of genetic information. They are the ones who form the basis of genetic code and also influence the metabolism by playing an important role in protein synthesis. It is through the sequence of nucleic acids that information is conveyed about hereditary elements. Basic structure of nucleic acids and nucleotides. Nucleic acids are polynucleotides, that is, long chain-like molecules composed of a series of nearly identical building blocks called nucleotides. Each nucleotide consists of a nitrogen-containing aromatic base attached to a pentose, 5-carbon sugar, which is in turn attached to a phosphate group. Each nucleic acid contains 4 of 5 possible nitrogen-containing bases: adenine, guanine, cytosine, thymine, and uracil. Adenine and guanine are categorized as purines, and cytosine, thymine, and uracil are collectively called as pyrimidines. It is established that all nucleic acids contain the bases adenine, cytosine, and guanine. Thymine, however, is found only in DNA, while uracil is found in RNA. There is a difference between pentose sugar in DNA, 2-deoxyribose, and the sugar in RNA, that is ribose, due to the absence of a hydroxyl group, that is a OH group, on the 2-carbon of the sugar ring. In the absence of an attached phosphate group, the sugar attached to one of the bases is known as a nucleoside. The phosphate group connects successive sugar residues by bridging the 5-hydroxyl group on one sugar to the 3-hydroxyl group of the next sugar in the chain. These nucleoside linkages are called phosphodiester bonds and are the same in RNA and DNA. Degradation and biosynthesis of nucleotides. Nucleotides are manufactured from readily available antecedents in the cell. The ribose phosphate portion of both purine and pyrimidine nucleotides is produced from glucose via the pathway of pentose phosphate. The 6-atom pyrimidine ring is synthesized first and subsequently attached to the ribose phosphate. The 2 rings in purines are synthesized while attached to the ribose phosphate during the amalgamation of adenine or guanine nucleosides. A nucleotide carrying a phosphate attached to the 5-carbon on the sugar is the end product in both the cases. Eventually, a specialized enzyme called kinase adds 2 phosphate groups using adenosine triphosphate that is ATP, as the phosphate donor to form ribonucleoside triphosphate, the immediate precursor of RNA. For DNA, the 2-hydroxyl group is removed from the ribonucleoside diphosphate to give deoxyribonucleoside diphosphate. An additional phosphate group from adenosine triphosphate is then added by another kinase to form a deoxyribonucleoside triphosphate, the immediate ancestor of DNA. RNA is constantly being made and broken down during the course of normal cell metabolism. The purine and pyrimidine residues are reused by a number of rescue pathways to make more genetic material. Purine is rescued in the form of the corresponding nucleotide, whereas pyrimidine is liberated as the nucleoside. Deoxyribonucleic acid, DNA. DNA is a polymer of the 4 nucleotides adenine, cytosine, guanine and thymine, which are joined through a backbone of alternating phosphate and deoxyribose sugar residues. These nitrogen-containing bases occur in complementary pairs as determined by their capability to form hydrogen bonds between them. 2 hydrogen bonds are required for adenine and thymine to form a pair, and guanine always pairs with cytosine through 3 hydrogen bonds. Chemical structure of DNA. A 3-dimensional structure of DNA was proposed by James D. Watson and Francis H.C. Crick in 1953. The basis of their proposition was the low-resolution X-ray crystallographic data and on the Erwin Chargaff's observation that in naturally occurring DNA, the amount of thymine equals the amount of adenine and the amount of guanine equals the amount of cytosine. It was Watson and Crick who postulated that 2 strands of polynucleotides coil around each other, forming a double helix. They also shared a Nobel Prize in 1962 for their efforts. The 2 strands, though identical, run in opposite directions as determined by the orientation of the 5-3 phosphodiester bond. Along the outside of the helix, the sugar phosphate chains run along and the bases lie on the inside, where they are linked to complementary bases on the other strand through hydrogen bonds. The right-handed form of the double helical structure of normal DNA is called the B helix. The helix makes one complete turn approximately every 10 base pairs. B-DNA has 2 chief channels, a wide major channel and narrow minor channels. The major channel witnesses the interaction of many proteins where they make sequence-specific contacts with the bases. Also, a few proteins are known to make contact via the minor channel. There are several structural variants of DNA. In aDNA, which forms under conditions of high salt concentration and minimal water, the base pairs are tilted and displaced towards the minor channel. Left-handed zDNA forms most readily in strands that contain sequences with alternating purines and pyrimidine. DNA can form triple helices when 2 strands containing runs of pyrimidines interact with a 3rd strand containing a run of purines. bDNA is generally depicted as a smooth helix. However, specific sequences of bases can twist the otherwise regular structure. For example, short tracts of adenine residues interspersed with short sections of general sequence result in a bent DNA molecule. Reversed base sequences, on the other hand, produce cruciform structures with 4-way junctions that are similar to recombination intermediates. The alternative structures are only established in laboratories and their significance is yet to be sought. Biological structure of DNA. DNA molecules which occur naturally can either be circular or linear. the genomes of single-celled bacteria and archaea, the prokaryotes, as well as the genomes of mitochondria and chloroplasts, which are certain functional structures within the cell, are circular molecules. Also, some bacteria and archaea have smaller circular DNA molecules called plasmids that typically contain only a few genes. Many plasmids are willingly transmitted from one cell to another. For a typical bacterium, the genome that determines all of the genes of the organism is a single contiguous circular molecule that contains 500,000 to 5 million base pairs. The genomes of most eukaryotes and some prokaryotes contain linear DNA molecules called chromosomes. For example, DNA of humans consists of 23 pairs of linear chromosomes containing 3 billion base pairs. DNA does not exist free in solution in all the cells. Rather, they are found as a protein-coated complex called chromatin. In prokaryotes, the loose coat of proteins on the DNA helps to shield the negative charge of the phosphodiester backbone. Chromatin also consists of proteins that control gene expression and determine the characteristic shape of chromosomes. In eukaryotes, a section of DNA between 140 and 200 base pairs long winds around a distinct set of 8 positively charged protons called a histone, forming a spherical structure called the nucleosome. Supplementary histones are wrapped by successive sections of DNA, forming a series of nucleosomes like beads on a string. Duplication and transcriptions of DNA are more complicated in eukaryotes because the nucleosome complexes have to be at least partially disintegrated for the processes to proceed effectively. Most prokaryote viruses contain linear genomes that typically are much shorter and contain only the genes necessary for viral propagation. Bacterial viruses called bacteriophages may comprise of both linear and circular forms of DNA. For instance, the genome of bacteriophage lambda, which infects the bacterium Escherichia coli, contains 48,502 base pairs and can exist as a linear molecule packaged in a protein coat. The DNA of phage lambda can also exist in a circular form and allows integration into the circular genome of the host bacterial cell. It is observed that both circular and linear genomes are found among eukaryotic viruses, but they most commonly use RNA as the genetic material. Biochemical properties of nucleic acids. Denaturation. It is the hydrogen bonding interactions between the complementary base pairs which hold together the strands of the DNA double helix. The hydrogen bonds are easily broken by heating DNA in solution, which allows the separation of the two strands, and this process is called denaturation or melting. The two strands may rejoin when the solution cools, changing the starting DNA duple, a process called renaturation or hybridization. These processes are the foundation of many important techniques for manipulating DNA. For example, a short piece of DNA called an oligonucleotide can be used to test whether a very long DNA sequence has the complementary sequence of the oligonucleotide embedded within it. Using hybridization, a single-stranded DNA molecule can capture complementary sequences from any source. Single strands from RNA can also rejoin. DNA and RNA single strands can form hybrid molecules that are even more stable than double-stranded DNA. The molecules form the basis of a technique that is used to purify and characterize messenger RNA, that is mRNA molecules corresponding to single genes. Ultraviolet absorption. The togetherness and the melting of DNA can be observed by measuring the absorption of ultraviolet, that is UV light, at a wavelength of 260 nanometers, billionths of a meter. When DNA is in a double-stranded structure, absorption is fairly weak, but when DNA is single-stranded, the opening of the stacks of the bases leads to an improvement of absorption called hyperchromicity. Therefore, the extent to which DNA is single-stranded or double-stranded can be determined by keeping a watch over the UV absorption. Chemical modification. There is a possibility of chemically modifying an already assembled DNA. It can be done deliberately by special enzymes called DNA methyltransferases and sometimes accidentally by oxidation, ionizing radiation, or the action of chemical carcinogens. DNA can also be cleaved and degraded by enzymes called nucleases. Methylation. The DNA consists of three types of natural methylation which have been found as per the scientific studies. The basic structures can be modified, as for instance cytosine can be modified either on the ring to form 5-methyl cytosine or on the exocyclic amino group to form N4-methyl cytosine. Adenine may be modified to form N6-methyl adenine. N4-methyl cytosine and N6-methyl adenine are found only in bacteria and archaea, whereas 5-methyl cytosine is widely distributed. Unique enzymes called DNA methyltransferases are responsible for this methylation. They identify specific sequences within the DNA molecule so that only a subset of the bases is modified. Other methylations of the bases or of the deoxyribose are sometimes induced by carcinogens. This is the usual cause of a mismatch in pairing of the bases during replication and has to be removed in order to avoid becoming mutagenic. There are many cellular functions to be performed by natural methylation. In bacteria and archaea, methylation forms an indispensable part of the immune system by protecting DNA molecules from fragmentation by restriction endonucleases. In some organisms, methylation helps to remove incorrect base sequences introduced during DNA replication. By marking the parental strand with a methyl group, a cellular mechanism known as the mismatch repair system differentiates between the newly replicated strand where the errors occur and the correct sequence on the template strand. In higher eukaryotes, 5-methyl cytosine controls many cellular phenomena by putting off DNA transcription. Methylation is also believed to signal imprinting, a process whereby some genes inherited from one parent are selectively inactivated. Correct methylation may also suppress or activate key genes that control embryonic movement. On the other hand, 5-methyl cytosine is potentially mutagenic because thymine produced during the methylation process converts cytosine guanine pairs to thymine adenine pairs. Methylation takes place selectively in mammals within the dinucleotide sequence. Cytosine guanine CG, a rare sequence, most probably because it has been lost by mutation. In many cancers, mutations are found in key genes at CG dinucleotides. Functions of nucleic acid and nucleotides. There are millions of cells that the human body is made of. These cells work in tandem to maintain the body's overall system and function. An individual cell, in turn, has its own set of processes intended to carry out necessary cell functions. Nucleic acid plays an important role in coordinating and maintaining individual cell processes throughout the body. Some of the functions are discussed at length below. Identification. A cell's genetic information is made up by the chemical compound called nucleic acid. Deoxyribonucleic acid DNA and ribonucleic acid RNA are the most common forms. The DNA double helix strands and RNA strands are connected to structural units or molecules called nucleotides. The sequence in which nucleotides are positioned on a DNA or RNA strand is the major factor that determines what code a nucleic acid holds. Each of these structural units contain a sugar, a phosphate group, and a base material. Both DNA and RNA combine with protein materials to carry out cell division and cell repair processes. Function. The structural formation of nucleic acids has cell repair and cell reproduction processes encoded within them. DNA strands are accountable for maintaining the cell's genetic information, while RNA strands work to coordinate cell processes according to these genetic instructions. RNA molecules are also occupied in manufacturing the proteins needed by the cell for normal function. In order to coordinate all of these processes, the RNA nucleic acids live in the cytoplasm that surrounds the nucleus, while DNA strands are located in the nucleus of the cell. Replication. The DNA nucleic acid has to imitate itself in order to reproduce daughter cells in the process of cell division. The daughter cells are identical in nature, identical in structure to the parent cell. There is a requirement that the parent cell has to split or break the bonds which connect the strands. Once broken, each strand can serve as a template or guideline for the making of a new set of strands. This process is called semi-conservative replication. Once completed, an identical DNA helix is formed. Transcription. The process in which DNA nucleic acids transfer the cell's genetic information into RNA materials is called transcription. In spirit, each DNA strand manufactures a corresponding RNA strand. 3 types of RNA are manufactured within this process. Messenger RNAs, mRNA, are designed to carry the genetic information received from the DNA strands. Ribosomal RNAs, rRNA, stay in the cell's cytoplasm and are responsible for decoding or translating the genetic instructions into cell processes. Transfer RNAs, that is tRNA, are accountable for gathering whatever amino acids are needed for protein synthesis. Translation. Translation is the process in which RNA molecules produce the proteins needed to sustain necessary cell functions. This is done by converting the genetic code contained in the messenger RNAs into amino acid strings, which is what the protein molecule consists of. This conversion process takes place within the ribosomes, which are located in the cell's cytoplasm. The proteins manufactured by ribosomes are then used to manufacture cell enzymes, the chemicals responsible for regulating cell processes. Conclusion. Let us now summarize the key concepts of this chapter. Nucleic acids are the polynucleotides having high molecular weight, the monomeric unit of which is a nucleotide. There are 2 types of nucleic acids, namely ribonucleic acid RNA and deoxyribonucleic acid DNA. DNA occurs in the nucleus as well as cell organelles like chloroplast and mitochondria, and RNA is found in the nucleus but mainly occurs in cytoplasm to carry out protein synthesis work. Transfer RNA, tRNA, messenger RNA, mRNA, ribosomal RNA, rRNA are the different types of RNAs. In animals, DNA forms a double helix while RNA is single-stranded. DNA and RNA also differ in the molecular units used to store genetic information, nucleotides. Nucleotides are the basic alphabets with which the genetic code is written. The DNA double helix has nucleotides on the inside, held in place by backbones on the outside made of sugars and phosphate groups. The double structure facilitates cell division. The basic functions of nucleic acids are transmission of hereditary characters, which is done by DNA, and synthesis of proteins, which is done by RNA. DNA is also the storehouse of genetic information, which serves as a blueprint for protein production elsewhere in the cell and responsible for the direct synthesis of RNA. Function of mRNA is to take the genetic message from RNA, and that of tRNA is to transfer the activated amino acids to the site of protein synthesis. Messenger RNA, that is mRNA, directly copies the genetic code from DNA before exiting the nucleus to create proteins outside. This enables DNA to remain behind, safe in the nucleus. Transfer RNA, tRNA, is nucleic acid that carries and attaches amino acids to the mRNA, which acts as a protein building template. The rRNA is mostly present in ribosomes and responsible for stability of mRNA. Each nucleotide can sit opposite only on its complement. For example, adenine can share a step in the spiral with thymine. Cytosine is the complement of guanine. If one nucleotide is damaged, gene repair mechanisms can identify the damaged nucleotide by its complementary nucleotide. tRNA is usually 70 to 90 nucleotides long, which is much shorter than mRNA. It comes in at least 20 varieties, corresponding to the 20 amino acids that are the building blocks of all proteins.